In this study, we identified the capsid and L protein interaction not merely regulate the capsid envelopment, but also took part in multiple steps of viral replication, which could also be affected by HBC linker sequence. In Figure 1, for the effect of L protein on viral intracellular application, the loss of L protein expression increased the rcDNA level in the wild-type capsid slightly due to the blocking of DNA Virion secretion. In contrast to the wild-type, the NTD L126A and the linker mutant S141A and L143I with unstable motor capsid and low to no Virion secretion showed decreased level of rcDNA in absence of L protein expression. This result indicates the influence of L protein on the cytoplasmic rcDNA could independent with DNA Virion secretion, and L protein could protect the rcDNA inside the unstable motor capsid from the degradation in the cytoplasm. In Figure 2, not only the unstable motor capsid, the LC mutant, which had 3 site mutations on the HBC linker with stable motor capsid, also showed decreased level of rcDNA in absence of L protein expression, which should relate to the enhancement of nuclear input by the LC mutations. While other 2 mature capsid stable mutant, E145D and AA, show no influence by the loss of L protein. And L protein not merely influenced the rcDNA in the mature capsid. Without the L protein expression, the level of double-strand DNA smear below the rcDNA also decreased in the wild-type and LC capsid. And during the loss of L protein expression, the wild-type and LC show increased of the protein-free rcDNA and cccDNA. But the other linker mutant show no influence on the level of protein-free DNAs. This result implied the HBC linker was critical for the interaction between the capsid and the L protein. In Figure 3, there was no surface protein expression in HEC293 cells, and the double-strand DNA smear could not be detected from the wild-type and LC capsid. The purified immature capsid of wild-type and LC from the HepG2 cells, without the influence of L protein, show similar co-DNA condition after the reverse transcription in vitro. This result implied the cytoplasmic co-DNA condition could be regulated by the L protein. In Figure 4, different with the genotype D L protein, the genotype AL protein could not restore the rcDNA of LC or the double-strand DNA smear in the wild-type and LC capsid. But both genotype A and genotype D L protein play similar role to restore the rcDNA and the double-strand DNA smear in the S141A capsid. In Figure 5, we purified the DNA Virion and detect the similar double-strand DNA smear inside the wild-type and LC Virion. It implied that the L-dependent DNA smear detected inside of the cell was indeed likely from the intracellular Virion, as the L protein could enhance the rcDNA and the double-strand DNA smear of S141A in the cytoplasm, which we introduced in the Figure 4. The intracellular Virion of S141A contains rcDNA were mostly returned in the cell, while the Virion contains the incomplete double-strand DNA prefer to secrete it out. This result implied the L protein and HBC linker work together to regulate the capsid envelopment and the time point of Virion secretion. The final model shows all the results we talk about, and the conclusion are in below. I can take any questions, and thank you for your attention.